It's lovely to be here this evening, and thanks very much for coming out on a bit of a windy Wednesday to hear about some open science, which we're going to start right now.
Oh, they're away, and Gatling got away brilliantly, and he's ahead of the field at the moment, and Bolt going very, and here comes Usain Bolt! Usain Bolt storming through, he takes it again, Blake gets the silver, 9.64! Oh, he's retained his title in the most emphatic way!
Of course, this is Usain Bolt, who broke the Olympic record when he got his second gold for this race. He wasn't actually in front at the beginning of the race. That was the American to his left. And he wasn't in front halfway through the race. That was his Jamaican friend, who's just over in lane number 4. But he managed to dip his head right at the exact moment to cross that finishing line with great style and to take that gold. And the reason that he was able to do that, apart from a little bit of training probably, is because he wasn't running in a tunnel. He could see what his competitors were doing. He could look left and right, and he knew exactly when to accelerate. He was also being watched, and these records often get broken at the Olympics because you're on the world stage. There's a huge amount of people watching what you're doing, and you really have to rise to the occasion and put your very best performance in. And when people are watching what you're doing, you're accountable to them too. And open scientists think that science should stop doing their work, or scientists should stop doing their work in tunnels and should really open it up for everybody to see, so that we can see what other people are doing, we can respond to other people's research, and we can really accelerate at the right time to try and make a difference or make a good decision when the time comes. And this is important for a number of reasons. Number one, transparency. By making science transparent, by putting all of your data out there in the open so anyone can see, you can help to build trust from the public, you can help to build trust from other scientists because they can scrutinize your research. And if you communicate that science well, both to your peers, and other scientists, and to the public, they can see what you're doing, they can understand the value of what you're doing. And this, in turn, leads to good decision-making, because the public can see how the government is spending their money on research. They can also make decisions that impact their lives, such as whether to vaccinate their children or whether they should care about climate change. So it's really important that as scientists, we are transparent, communicative, and that we open up our research to anybody who is interested, and even those who might not think they're so interested. Now, scientists have been sharing their research in a variety of ways for centuries. There were often meetings where scientists would gather to explain their theories to each other. Thanks to places like the Royal Institution, these conversations stopped being just between scientists and have been projected outwards to the public, which is really important. But this is an important document because it's from 1665, and this is the first academic journal. So this is the first time that a collection of scientific theories were published and disseminated in one volume. And before this, they'd been kind of shared in different ways. People didn't know exactly what was going on. And in later issues, the idea of peer review was introduced. So this is the idea of something being submitted for publication, a group of experts checking to see that that science is rigorous, that it's been well done, before it gets published as the truth, or the truth so far in the scientific picture. And this is the way that most science is shared today. It is the main currency for scientists. You have to publish or you perish. You have to publish in good places. I won't describe what good means because that's a whole other presentation, but you have to publish in places that people will read your research so that you can keep your job, so that you can get money to do more research, and so that you can get promoted if you're interested in that. But there are a few problems with this model as far as I can see, and as far as others. It's not just me who thinks this. One of the things when we look at this industry, the publishing industry, it's a pretty good industry. It has a total global revenue of over £19 billion. And when you look at the margins, the profits that are made, this is from one publishing company, Elsevier, from I think 2011. They had a 36% margin on these profits, which was higher than Apple or Amazon or Google. So it's a pretty good industry for making money. And that's because it's got a really unusual setup. So a normal magazine style publication makes about 10 to 15% profit, but that's because you have to pay a lot of people along the way. Whereas in scientific publishing, what you're doing is you're relying on a scientist to produce some excellent work thanks to funding from the taxpayer. And then when she's got a really exciting result to publish, she sends it to a publisher to see if they want to print that. And before it goes out, the very important peer review stage happens. This happens by another scientist, in fact, at least 3 scientists, who do this for free. So they peer review the work and decide whether or not it's suitable for publication. And if all 3 of your reviewers agree that it's good enough for publication, it gets sent back to the publisher. And then the scientists, as a collective, have to pay money to read all of the journals. So essentially, the public is paying twice for this model because they're paying for the subscriptions for the university and for the research. So it's a model that is really quite unusual, and I'm a little bit surprised that it's still the model that's in operation today. This is something that's been changed, and many of you will have heard about open access publishing. This is a great a shift in the way that data and scientific information is being shared. And there are many different types of open access, which I don't have time to go into today. But essentially, the idea is that that publication, that finished product, will be accessible to everyone at some stage in the future, so it's not locked behind a paywall. This is really important because that means that the public can read the research that they've paid for. It's also really important that it's communicated in a way that the public can understand that research rather than it just being written in very technical language. It's also incredibly important for universities and institutions in developing countries who simply do not have enough money to pay the subscriptions for these journals. But sometimes open access isn't quite far enough because it's still work that's being done in tunnels that becomes public at the end of the tunnel. It doesn't, it's not a transparent process the whole way through, so you have to wait quite a while to see the results of that studying. So we're going to play a game of Family Fortunes to sort of demonstrate this. So if I were to give you the topic of a UK soap, would anyone like to suggest a UK soap to me? Brookside. Anyone else? EastEnders. Thank you. Anyone else? Okay. We'll skip on from now. So we have, as the first answer, Coronation Street, EastEnders. Brookside didn't quite get up there, but I think it's probably at number 4. But the demonstration I was trying to make there is the fact that as humans, okay, UK soaps might be a little bit less complicated than, splitting the atom. But as humans, we think about things in similar ways. So if as scientists we're thinking about things in similar ways, but we don't know what other researchers are doing, we can be duplicating efforts. And that's bad enough when people are doing the same work and they're racing to get to the prize from the same research. But if they're doing the same research and it doesn't work out, then that means that lots of time and money is wasted. And that's particularly important when it comes to research that can influence or improve human health. So drugs, the discovery of drugs. And finding new medicines, which are incredibly important for all of us, is a really difficult and expensive process. It's a very tricky thing to do. It costs a lot of money, and it takes a lot of time. On average, it takes about 12 years to get a drug from the discovery right to the pharmacy, and it costs about $2.6 billion US. And because of this, there's a whole load of secrecy involved because there are huge profits at stake. So unsurprisingly, people need to protect the investments of shareholders. Now, when we think about that figure, that 2.6 billion figure, a little bit further, and we consider that 25% of that is spent on marketing, does anyone want to hazard a guess as to how much is spent on research? 10%. So we'll play another game, higher or lower? Lower, okay. Well, we've had 2 colds. So it's actually about 1.3%. So there's a huge disparity in those numbers. And this is problematic for all of us because you would hope that lots of money would be going into research for the drugs that we all need or may need in the future. But it's especially problematic for drugs that are needed for developing countries. So in recent years, we've had an outbreak of Ebola, we've had an outbreak of Zika virus, and people looked on in horror when they discovered that these viruses have been known for a long time, and they're genuinely surprised that there weren't cures to treat these viruses. But when you have an industry for which there's no market incentive to develop a drug, then it shouldn't be surprising that we don't have medicines to treat these diseases. And this is something that's been thought about at the very highest levels. This is from a speech by Dr. Margaret Chan, who was then the World Health Organization Director General. And she flagged this issue in some speeches to the World Health Organization. Another disease that is particularly prevalent in developing countries is malaria. You can see from this map that it predominantly affects the world's poor. And so this is one such disease that really requires new medicines for markets that can't really pay very much for the medicines that are needed. This is the female Anopheles mosquito, who's responsible for the transmission of malaria when she takes her blood meal, but more on that a little bit later. And in 2015, there were 212 million cases of malaria and 429,000 deaths. And the vast majority of those were in sub-Saharan Africa, and they were children under the age of 5. And given that malaria is an entirely treatable and preventable disease, those numbers just don't cut it. We need to think about how we can do something to reduce the disease burden. So we're going to have a little pause now, where we're going to play another little game, where I'm going to give you 2 truths and a lie about malaria. So we'll start off with Jurassic Park. So some of you might have seen this film. It did quite well. And my first fact is that, or is it a fact, that the origins of malaria date back to the dinosaurs. So there's a rich history of malaria that's been charted by several Nobel Prizes across the centuries. In 1880, there was a Nobel Prize for Charles Louis Alphonse Laveran because he was the first person to see malaria parasites in the blood of a patient. And before the time of Twitter, in the Roman era, there were huge outbreaks of malaria, which are by some attributed to the decline of the Roman Empire because a lot of the soldiers got very sick. But before that, a little while before that, some researchers think that the origins of malaria are actually found in this particular amber fossil, and that an ancient form of malaria might be even older from this biting midge, might be about 100 million years old. So that's fact number 1. Fact number 2, so this is gin and tonic. Some of you may have experienced this beverage before. So my fact number 2 is that drinking 3 to 4 gin is not necessary, but the tonic water with lime and lemon daily is as effective as preventing malaria as prescriptive medicine. So I'll give you the facts based on this. So if you were to drink 3 to 6 glasses per day with an average volume of 350 millilitres, they would have enough quinine, which is an anti-malarial, to eradicate the disease in your system, thanks to the presence of lime or lemon, which contains citric acid, which has a synergistic effect and makes quinine more bioavailable in your body, causing you to be able to deal with the malaria parasite. So, number 3. This fact is that the frontline treatment for malaria today comes from Chinese medicine. So this is artemisinin, which is also known as the sweet wormwood. So there's another sweet wormwood. And this molecule is found in these leaves and is currently the best cure we have for malaria today. And it was extracted a very long time ago. This is a Mao-era version of a very ancient Chinese medical book. And in the 5th column, it says, a handful of sweet wormwood immersed with 2 litres of water, wring out the juice and drink it all. And this was a treatment for fever. So we've got malaria, could be as old as the dinosaurs. Gin and tonic is as good as prescription medicine for the treatment of malaria. And the best medicine for malaria comes from a Chinese medicine. So can I have hands up for the one you think is false? So if you think number 1 is false, the dinosaur one. If you think number 2 is false. If you think number 3 is false. Okay. I think the 2s, they did just about pip everyone to the post, so that was the lie. I use the word synergistic, and sometimes that sounds a bit technical, but if you wanted to have a therapeutic dose of quinine from purely gin and tonic, because the Federal Drug Administration's upper limit for quinine in tonic water is 83 milligrams per litre, and you need about just over 2 grams of quinine for a day's treatment, you'd have to drink 25 litres of tonic water for about 5 days. So best to stick to the medicine. So back to artemisinin. This beautiful molecule is indeed from the sweet wormwood, and it's been known to Chinese medicine for thousands of years. It was for the treatment of fever. It's a particularly interesting molecule as a chemist because if you look at that top left-hand ring, there are 2 oxygens that are singly bonded to each other, and that means explosion to an organic chemist. So I think if people had known the structure of that molecule before it was used as a therapeutic, it probably wouldn't have got that far because that means danger to the eyes of an organic chemist. Now, in 2015, Professor Tu Yu won or shared part of the Nobel Prize for her work in understanding the structure and the effects of this medicine. And artemisinin and other related molecules in combination with other medicines are the very best treatment that we have for malaria today. So there's a lot of gold in those books. The problem is, unfortunately, that resistance to this type of therapy is spreading. Some of you may have heard these so-called reports of super malaria. The only thing that's super about it is that it's resistant to every single medicine that's available. And this is a very worrying prospect because malaria cases have actually been reduced by about 22% between the year 2000 and the year 2015. But if this resistance spread, which is looking increasingly likely, those cases in sub-Saharan Africa are going to rise extremely quickly. So we need a new medicine to treat malaria, and we need it really quickly. But when you think about how much it costs to make a new drug, and you think about how long it takes, we might have to start thinking about a different model for the discovery of these medicines. And this is where open source drug discovery comes in. So most of you today, in fact, I'd hazard a guess as to all of you will have used some sort of open source software. If you've done a search on your phone, you might have used an open source program such as Wikipedia that's put together by the collective. Now, software developers are ahead of everybody in this game. They have been putting out code for a long time now and allowing other people to edit that code to use it for whatever purpose they'd like to in order to improve it for everybody. And this is the idea of open science, that we would put all of our things into the public domain and people can use it and improve on it and use it for whatever they want to. So the idea of an open source drug discovery project for malaria was first thought of and set up back in 2011 by Associate Professor Matthew Todd, who's my colleague at the University of Sydney. And he founded the Open Source Malaria Consortium with funding from the Medicines for Malaria Venture, who are the global gatekeepers of all malaria research, and a linkage grant from the Australian government. And I joined him about 9 months after he started this project as his postdoc, working in the lab to make some medicines as a chemist, which is my job. Now, the way that we do our research is we put everything out in the open. And one of the analogies for this, I've just seen an open lab just downstairs, so I encourage you to go and have a look at that. But one of the analogies is between a restaurant where we can see what the chefs are doing. Because if you can see that the chefs have washed their hands and they're not picking their noses and that they're cooking things that look clean and tasty, it gives you confidence in the food you're about to eat. And we hope that that's the same with the science that we do. So we have some laws that Matt set up, which sound rather grand, but essentially all of these rules or principles for the project allow us to share things. They stop any barriers for keeping things secret. And these are really important things for us to adhere to in the project. So we won't patent anything, for example. We try not to have conversations on email, but rather to have them on discussion forums. And it requires a little bit of adaptation for people to be involved in this sort of process. But thankfully, lots of people have joined in. There have been researchers from all over the world who've joined the Open Source Malaria Consortium, because drug discovery does not just need chemists, but needs biologists, it needs cheminformaticians, it needs people who can analyse data. So it's really a big collective approach to this science, as pretty much all science is. So as you see the Nobel Prizes coming out, only 3 people can get the Nobel Prize. But there'll be thousands of people who've contributed to those awards, so it's always good to remember that. Now, one of the kind of the main important part of this project, and for the scientists in the room, this might be new to some of you or not to others, but is that the lab notebook is actually online. Leonardo da Vinci kept some beautiful lab notebooks a few hundred years ago that I've never quite managed to live up to. But one of the problems is that most scientists are still keeping paper notebooks. And really, we need to start preserving our record electronically. And we did this in real time. So we actually write up our results, our experiments, as we're doing them at the lab bench. That can be a little bit scary. So if you drop your reaction flask, you have to tell your lab notebook that happened, rather than just working for a few extra hours over the weekend. And if you put the wrong reagent in, you have to say that too. I'm not saying I know anyone who's done that before. We also share all of our data, both in a processed form and in a raw data form. And this is an analogy here between potatoes and chips. So chips are wonderful, but potatoes are even better, because with potatoes, you can make chips, you can make a massaman curry, you can make a cottage pie, you can make whatever you want. And this is what we need with data. We need to have the processed form, the chips that are easy to look at and easy to digest, but we also need to have the raw data that can be manipulated into anything we'd like it to be. We also try and communicate the project, and we do this through a wiki, which is human readable, and collect our data in a way that the public could read what we're doing, other scientists can read what we're doing, because sometimes we make this sweeping statement of scientists talking to one another as though a chemist understands everything that a virologist is saying. And sometimes, you have to meet somewhere in the middle and find a way to communicate those ideas too. And in terms of decision making, all of this is done on online forums. This is GitHub that's usually used for sharing code, but we use it as a project management tool, and we use it to make all of our decisions and assign different jobs to people who are within the team, and to make those decisions in the open. Now, a bit more about malaria. So one of the reasons that malaria is such a tricky disease to treat is because it's a parasitic infection that has many different life stages. So it requires both the host and the vector, the person or the animal and the mosquito, for transmission. And you can see that all of these little blue diagrams are a different life stage of this parasite. So when a mosquito bites a person, it's always a female mosquito because she needs blood to nourish her eggs. She actually injects the parasite through her salivary glands, and then the parasite takes hold by basically burying into human red blood cells and living in there, and it travels around the body, developing into different life stages. It can hide in the liver. This is why we sometimes have dormant malaria that can come back a little bit later. Then there's a sexual stage. A mosquito comes back to take a bite, takes up these gametocytes, which can then reproduce in her stomach, and then the cycle begins again. So because there's so many different stages, if you kill one stage but you haven't got rid of all of the next stage, it's very difficult to really combat this parasite and to kill it completely. But you can when you work as a team. So this paper is a really important paper for our research project. It actually came from the pharmaceutical company GSK. And this was a groundbreaking project or a paper, because what GSK did is they took their 2 million compounds, because they have huge compound libraries because they've done so much drug discovery over the years, and they screened them in a high throughput screen against the malaria parasite and found about 13,500 molecules that were active against the malaria parasite. And rather than keeping those secret, they put all of that data into the public domain. And this was a great thing because many other papers have sprung from this, and people have looked at that data and really honed in on which of those molecules are particularly promising targets for chemists to make, for biologists to test, and for the whole process of research to continue. And this really shows us just how important organic chemists are, as well as other scientists. I'm a little bit biased, but most of the past cures for malaria have been natural molecules. So they've been things that have been derived from plants and found in nature. And organic chemists can make natural products. We can recreate the molecules that nature makes. And when we recreate them, they are exactly the same as the natural compound. There's no difference between those molecules. They're exactly the same. But we can also make molecules that nature has never made before, which gives us, I wouldn't say an advantage over nature, but it means that we can try to kill natural things, such as a parasite, with an unnatural or a synthetic molecule. And many of the drugs in this paper are unnatural. That sounds a bit dodgy, but they're synthetic molecules. They're things that have been made rather than made by nature, made by man rather than nature. So we've had a huge team of researchers who've been involved in this project around the world. But because we're open, I'm going to really focus on some of the more interesting collaborations that have occurred because we can share our data in this talk. So a few years ago now, it was a bit of a rainy afternoon in Sydney, which always makes Australians very grumpy. And I realised that I wasn't going to have enough time to finish making the molecules that had been assigned to me for the next meeting. So I made a wanted poster. You can see why I'm a chemist rather than a graphic designer. And I posted it on Twitter, and much to my delight, somebody replied that by the time I'd got up the next morning, Patrick Thompson from the University of Edinburgh said, I can make those molecules with some students in my laboratory. Now, I'd never met Patrick. He'd never met me. We could have been, two random people on Twitter. I guess we are to some people. But because Patrick's research team were contributing to the same electronic notebook and uploading all of their data, all of their potatoes as well as their chips, we could see what they were doing in real time and check that they'd really made those molecules. And Patrick sent those molecules away for testing at the University of Dundee. All of them were inactive. That's really, really important because so often in drug discovery projects or in science in general, we lose the things that didn't work. And we need to publish the things that don't work so that nobody else spends time repeating that work. Or if they repeat it and find out we were wrong, even better, we can have a discussion about what's going on. So importantly as well, lots of the work from the first few series of compounds that came out of that paper from GSK were published in one of these traditional academic journals, the Currency for Scientists. We were really fortunate to be able to publish work that had already been published, which is a very difficult thing to do in an academic journal, and to publish in an open access journal so it's completely accessible, and to link to data that wasn't finished, so to link to spreadsheets that are still being populated with more data. So that was really important for us too. Now, the current set of molecules that we're working on at the moment are the triazolopyrazines. They're called the triazolopyrazines because that embolded black core is a triazolopyrazine. And what we're doing is we're basically changing the blue and green blobs at the moment for different structural groups to try and see if we can find the perfect or the best molecule for killing malaria. And there's a few reasons why this particular type of molecule are exciting. One of the reasons is that these two molecules shown on the right can kill the malaria parasite in mice. And that's really important because often, we always do our first tests in dishes, in Petri dishes, but trying to kill something in a living system without killing the living system can be a challenge. So it's good to know that these work really well in mice too. There's also another reason that we're particularly interested in them, and this is very interesting from a scientific perspective, but also important for resistance potentially. We think that these molecules kill the malaria parasite in a way that none of the drugs that are currently on the market kill the malaria parasite. And that could be important for resistance because if the parasite has developed resistance to a particular form of attack, then if we could find another way in, they might be good at killing those resistant strains. It's also interesting because we just want to know how this actually works. And because of collaboration with Professor Kieran Kirk's group at the ANU, who are a fabulous research team, we think that what's happening is that in some way, our molecules are blocking an ion transporter that is connected between the parasite in the red blood cell. So rather confusingly, the blue circle is a red blood cell, and the yellow circle is a parasite. And the parasite is feeding off this red blood cell, getting its nutrients from the red blood cell. But if you block this channel, you can no longer have this exchange of sodium cations and H+ cations. And the concentration of sodium in the parasite begins to get larger and larger, and then eventually the parasite swells and bursts, and it's gone after that, it's dead. So the other thing that's pretty interesting about this for us is that we have no idea what the shape or the structure of that channel is. Often when you're designing something, you know the shape of the biological target. Think about that as a keyhole. So you design a key that will fit into that hole. We don't know what the shape of the hole is because it's too tricky to crystallize this at the moment and get a snapshot of that structure. So we're trying to guess the shape of the hole by looking at the molecules that are active. And there are other research groups who have very, very different shaped molecules that are also active, which is very curious at the moment. And because we're an open project and we wanted to know if we could build a model for this, we had an open competition online and asked computational modelers if they wanted to be involved in this project. And we announced the tie of the two winners, I think a week or two ago. And we have some data now to try and help us to predict which molecules to make next, rather than just making things that we think will work. Hopefully this is going to help us to be better equipped. So I'm now going to switch back to my undergraduate days as an organic chemistry student at the University of Leeds. My favourite thing in the week was my organic chemistry labs, and I used to spend quite a long time in them, making different crystals, trying to see if we could make molecules that didn't exist at the beginning of the day. And then we'd spend a few weeks making them, characterising them, and then giving them in to be looked at by our head of lab, who is called Dr. Sparks, which I just think is the best name for a chemistry lab supervisor. But the thing that used to break my heart every single time I handed them in after I'd got my marks back was that I saw that all of those samples ended up in the bin. So I used to think, why are we making things that the university isn't using? Why don't we make something that's useful? And that's something that stayed with me through my postdoc, and I was really keen to see if we could get undergraduate students involved in making molecules that are of research importance or value or significance. And I've managed to do this with a first-year team at the University of Sydney. And there's a few barriers to overcome when you're asking a group of first-year students to share things, because especially when they're really gifted students, it can be a bit intimidating to share things if you're not quite sure if you're right, or if you're not quite sure if something's worked out. And also to tell them that we're doing real research in the first-year lab, and that sometimes our experiments won't work. And that is extremely alarming for first-year undergraduate students, who I think, until about 4 weeks in, don't really believe that I haven't tried everything out. And some of them are thinking, why hasn't she tried everything out? But we're trying to give a real research experience and to do something that actually matters. So at Sydney, a group of 60 undergraduates each year for the last 3 years have worked on the synthesis of triazolopyrazines. And in the first year, so they made 4 as a class. They were in 4 different groups, so that if anything went a little bit wrong, they could borrow some material from each other, or they could compare their data to see if things looked like everyone's was working out. And they actually made a really active molecule, one of the most active molecules that we've made as part of the research program so far. This one shown in the yellow box. And in the second year, we looked at changing the group on the right-hand side, and we found some other active molecules, not quite as good as this one, but some still promising and important information for the project. And this year, I went a little bit crazy and went double or quits and tried to see if we could make 8 molecules as a class. And the most exciting thing that's happened so far in this program happened this year, and that was that Team Ed didn't manage to make any of their molecules. And for me, that was a real test that the project was working, because we tried something that we didn't know whether it was going to work in the lab or not. And Ed is a wonderful PhD on the project, so he's probably the most experienced demonstrator he is the most experienced demonstrator for this project within the group, but his team didn't manage to make the molecule, and the students could really see that a life of research involves a lot of things that don't work, and it's important to have that experience quite early on. So now I come to another part of the project today, and this is the idea of getting high school students to make drugs. Now, if you're lucky, when you mention the first idea of having high school students making drugs, people think about the rather affable and talented Walter White. But if you're not so lucky, they think about the notorious crime lord of Heisenberg. But our project isn't Breaking Bad. We're not trying to make illicit substances in the lab. We call ours Breaking Good, because we're trying to make some molecules that hopefully of some value to society. And the team at Sydney Grammar that I've been working with have been working on organic synthesis before and after school. First of all, we're just going to have a little comparison between methamphetamine, or crystal meth, which is on the left, and one of our molecules in the lab. They look a little bit different to begin with, but you'll see after a little while that they have a number of common structural features. So they all have these benzene rings. There's some information on the structural determination of this downstairs. You can go and look at that sometime too. They both have a nitrogen that's bonded to a hydrogen, also bonded to a carbon. And there are other similarities in there. They're basically made up of nitrogen, carbon, and hydrogen, these molecules, with a cheeky oxygen on the right-hand side. So if you have the skills to make an illicit drug, you also have the skills to make a medicine. So it's important that we try and get people to be using chemistry for good rather than for bad, and teaching these valuable skills early on so that people know what chemists do. Because often you don't find out about synthetic chemistry until you've started a chemistry degree. So in the first year of this project, the Sydney Grammar students worked on the synthesis of those triazolar pyrazine molecules, and they got as far as the last step in the synthesis. And we've had a rather lovely story in the lab where one of the current honours student in Matt's lab, he actually mentored those students from afar. He was their online mentor while they were working on those molecules. And they didn't quite get to the last stage, but he's now taken the molecule that they made and taken it to the next stage and got a beautiful crystal structure of those molecules and submitted it for testing. So the student worked on the malarial molecules for a little while, and then in 2015 I was thinking about a new project for the students and thinking about what they might make, when a molecule that unites all four of these images received some attention in the press. And the people on this slide, first of all, this is the Sydney Grammar School logo at the bottom. Above that is Gertrude Lyon, another Nobel Prize winning female. She won the Nobel Prize, for a few things, but really for looking at this idea of rational drug design, this idea of designing a molecule that could specifically be for the treatment of a disease. So she was a wonderful scientist. You probably know Hillary Clinton. She's not the President of the United States of America. And in the bottom right, we have Martin Shkreli, who's he was the then CEO of Turing Pharmaceuticals. And Martin's really the reason that this molecule came into the public knowledge or into the media, because Turing Pharmaceuticals bought the market rights for this compound, which is daraprim, or pyrimethamine, which was originally invented by Gertrude's team for the treatment of malaria, and it's still used in some cases today, but it's mainly used for the treatment of another parasite, which is toxoplasmosis. And one day in September 2015, Turing Pharmaceuticals hiked the price of this molecule from $13.50 a pill to $750 a pill overnight. Now, this medicine is listed as essential by the World Health Organization because although toxoplasmosis is largely innocuous, and on average about 30% of us here will be carrying this parasite, if you're pregnant or if you're living with an immunocompromising condition such as HIV or AIDS, it can be really dangerous. So we thought, why don't we see if the students could make this molecule in their high school laboratory? We could have a really good organic chemistry project, and we could also have some good discussions about the ethics of drug pricing and drug discovery. And the school students did just this. So they made this this active ingredient in 3 chemical transformations. It took them about a year of doing research before and after school, but they're very busy students, so they do a lot of other things too. And they got the initial route from Wikipedia, but we had to change the second step, and they did this with their wonderful teacher, Dr. Malcolm Binns and Dr. Erin Sheridan, because the second step in the patent involved the use of a reagent which is extremely explosive and probably wasn't suitable for use in a high school. So they managed to get around that and use something that was far less dangerous. And the students made just under 5 grams of the active ingredient, which was worth, and I use the word worth loosely, over 130,000 US dollars. Of course, it's not worth that at all. In Australia, you can buy 50 tablets for $12.99. So there's really no reason for this molecule to have been hiked. The patent expired about 40 years ago. And so we could have some really interesting discussions with the school students about why this drug was so expensive in the States. And we received some press coverage for that story, which was a little bit overwhelming at the time, but quite exciting to see chemistry making the news, because sometimes it's a little bit forgotten. This is a timeline of all the different universities and our school that are involved in the open source malaria project. I've mainly talked about Sydney schools today, but it's really important that I mention the first team who joined in were Lawrence University in Appleton, Wisconsin. And we also work with Haverford College and MCPHS University in the States, Massey University in New Zealand, Reading in the UK, Griffith in Australia, and now the University of Birmingham, and many of the universities are interested in joining this programme. We're also looking to grow the schools project and find ways to bring real research projects to schools in the years to come. So that's Dylan holding 150 grams worth of this compound in the local Sydney paper. So the idea is to mobilise some of the Jessies and the Heisenbergs from around the world, and rather than getting them to break bad, to see if they can do something useful. They can make molecules that wouldn't have otherwise been made and break good. We're also working on something we won a small grant for from Hitachi last year, and this is the Science Introduction Robot. And you will see that the acronym is actually CINDER, and the idea is that this is a Tinder for scientists. So based on your algorithms, it will find scientists who are working on similar projects, and you unite those scientists so they can swipe right on each other. So rather than finding your lifelong partner, which is what Tinder's for, you can find a lifelong collaboration with a scientist. So we're working on that at the moment, and we hope to introduce this into an electronic notebook at some point. By we, I say we in a very collective sense, because there are some very talented cheminformaticians and coders who are involved in the project. So I think I'll leave it there. These are some information. This slide has some information about where you can go to find out further information for the project. I haven't acknowledged everybody who's involved in this project because there are so many people. So I encourage you to go and have a look at the open source malaria website so that you can see the names and faces of people who are involved. Of course, I mentioned Matt Todd, who is the founder. You can also check out the Breaking Good project. We also have projects in open source TB, tuberculosis, open source mycetoma, and a general open source pharma consortium that Matt Todd is a part of, thinking about a new way of making and finding medicines that can maybe challenge the traditional model. And we were recently featured on The Daily Show in a rather fun sketch that you might want to take a look at later. So thank you very much.